Challenges and Future Perspectives, being organized by Sun Institute of Pharmaceutical Education and Research, Kakupalli, Nellore, Andhra Pradesh. Today, eminent personality Dr. N. Mahadevan, M.Pharm, PhD, Professor, Department of Pharmacognosy, are admitted to deliver his efficient lecture. Hearty welcome and thank you for joining us, respected sir. Before going to today's session, a small introduction about our college. Sun College of Pharmaceutical Education and Research is established by the Sri Lakshmi Venkateswara Society. The founder and chairman of the society is Dr. A. Dinakar Reddy Garu. The college affiliated to JNTU Anantapur, Andhra Pradesh and approved by ASCT and PCI. It started a Bachelor of Pharmacy in 2009 and M.Pharmacy with specializations of pharmaceutics, pharmaceutical analysis, pharmaceutics with drug regulatory affairs in 2013. PharmD program also have been started in 2014. Now I diploma in pharmacy also have been approved. Thank you, sir. It's my privilege to introduce our college. Now I request Dr. M. Shekhar, sir, Principal of Sun Institute of Pharmaceutical Education and Research, to introduce today's speaker, Dr. N. Mahadevan, sir. Thank you, sir. Over to you, sir.
Good afternoon, everybody. I'm Dr. Shekhar, Principal of Sun Institute of Pharmaceutical Sciences. Today we have our speaker, Dr. Mahadevan. He has got vast experience of more than 25 years of experience in the field of pharmacy teaching. He has graduated B.Pharm, M.Pharm, and doctorate in pharmacy, altogether from the prestigious MGR Medical University. He hails from Ooty, so he has studied and has worked in JSS College of Pharmacy for beginning in his career. Later on, he has worked in different colleges. In JKK College of Pharmacy, he had worked for a long period also there also, and he was been as a principal for different institutions. He has published around 70 to 80 international papers, and he got national papers also. He has been the reviewer for many journals. He had been attended many seminars, poster presentations. He got a vast experience in pharmaceutical teaching and research. He had been so helpful for many of the students. Presently, he is working at King College University, Abba, which is the very coolest place of Kingdom of Saudi Arabia. So
Yes,
as he hail from Ooty, he has chosen the second Ooty in Kingdom of Saudi Arabia. So with this, I ask our Mahadevan to take on to the today's session. Thank you, sir. Thank you all students and invitees. Thank you very much. Mic is over to Dr. Mahadevan.
Thank you, sir. Good afternoon to all of you. First of all, I would like to thank the principal of Sun Institute, Dr. Shekhar, for introducing me with nice words. Thank you, sir. I also thank the organizers, the management for giving an opportunity to the students and staffs regarding by conducting the international seminars. So that will be really helpful to the students and the staffs. Sunil, are you able to see my screen? Sunil, hello. 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 Hello.
Sir, it is visible, sir.
Can you hear me?
Screen is visible, sir.
Can you hear me?
Yes, sir.
Okay, no problem. I'll start. So, dear attendees, for the next 1 hour or more than 1 hour, I'll be talking about biosimilars. the current status and future perspective. So attendees, I think you are mainly the students and some of you are the staff members. And many students are hearing this one for the first time, I believe so. So I'll be starting my presentation with the basics, starting with a small introduction. Dr. Sunil, some problem? Always I'm getting some, hello. Hello, Sunil. Hello. 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 Can you hear me? Hello. 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 Can you hear me?
Sunil sir.
Hello, sir, Mahadevan sir.
Can you hear me?
Yes, sir. Yes, sir. Yes, sir.
No problem. No problem. But somebody,
problem.
at least one person should be like means you should not mute yourself, because sometimes I will
Okay.
ask some problem
Okay, sir.
comes unable to hear something like you have to answer.
Okay, sir. Okay, sir. No.
Please. Okay, dear attendees, sometimes you'll be hearing this word for the first time. So I'll be giving the introduction of the biosimilars. So under the introduction, I'll be teaching about the different, the names of the, new names. That means what is the meaning of reference product? What do you mean by interchangeable product? What is biobetters? So these are different terms that should be related to the biosimilars, and one must know about these different terminologies. Later on, I'll be moving to the advantages of the biosimilars. So certainly the biosimilar has certain advantages over the conventional drug. So I'll be talking about the advantages of the biosimilars. Then of course, I have to talk about the current status of biosimilars. So in this case, what about the current status in USA or European unions or in India or any other throughout the world? So what is the current status that I'm going to talk in this webinar? Then the important point in this webinar is the development of the biosimilar. So how biosimilars develop? So there's a process, of course, very difficult process. So I'm going to explain this development process in a short way. And later on, there are a lot of challenges are there with the biosimilar development. So what are those challenges? How that can be overcome? So all these we are going to, I'm going to talk in this webinar. Then safety consideration and future perspectives that I'm going to talk. Then later on, I'll be concluding my seminar with the safety consideration. So dear attendees, just I want to tell you the meaning of biosimilars. So what do you mean by biosimilars? Biosimilars are the biological drugs that are similar to and cheaper than the other biological drugs. That means biosimilar and biological drugs, one and same only. Biosimilar is a copy of the original biological drug. Again, I tell you, biosimilars are the biological drug that are similar to biological drug and cheaper than the biological drug. Means the biological drug which is already available in the market, that is known as the reference product. That means biosimilars are biological product which is produced based on a reference drug. So reference drug, or you can say reference original biological drug. So this was already available in the market, which was already approved in the market. So based on this, we will have a copy. So that copy is known as biosimilar drug. So biological drug or biosimilar drug. So these are created in the living cells. So these are not produced chemically. That means these are cannot be synthesized like chemical drugs, and they're generics. So these are not generics. These are biosimilars, are not generics. These are not copies of the biological drug. Why it is not copied, that I am going to tell you in the next slide. Biosimilars are officially approved version of the original innovator product and can be manufactured when the original product patents expires. So attendees, just to make a note, biosimilar drug some disturbances in my screen. Sometimes something is popping out, so that is the reason. Okay, so biological drugs and biosimilars both are produced from the living cells, and biosimilars are the copy of the original drug. So this original drug that can be prepared, that can be manufactured once the patent of the original product expires. So there's expiry of the original product. So after the expiry of the original product, then this original product may be copied. That means biosimilar product may be produced. So this is a big condition for preparing the biosimilar product. So that the biosimilar product is not regarded as a generic copy of a biological medicine. So why? So there is a reason. So the reason is that this is because of the natural variability and more complex manufacturing of biological medicine that do not allow an exact replication of the molecular micro heterogeneity. That means what? We cannot prepare an exact copy of the biological drug. So if it is not exact copy, it is not the generic drug. So what it is and why it is? Why we are unable to prepare a copy of the biological drug? So there are a lot of reasons. So as I told you, biological drugs are the complex drug. That's one reason. So because of that reason, during the manufacturing, there will be natural variability. That means addition of certain groups, or sometimes it may undergo some stereochemical changes. The size may change, the shape may change, some changes that may takes place that is uncontrollable because of the complex manufacturing process. So because of that reason, we are unable to prepare a exact copy of the biosimilar product. So that is the reason we call this drug as a biological drug, or biosimilar drug, in fact. So what do you mean by biological drug? So I want to give you some example with this slide. So already you know the meaning of biological drug. That means these are the large complex molecule than the chemical drugs. So when compared with chemical drugs, these are large and complex molecules. So that already you know, there is no problem at all. And you also know that the biological drugs that is produced through a biotechnological way in a living system. So we can use either microorganisms or any other cells, and that's very difficult to characterize. So these are the different properties of the biological drug. So it's very, very difficult to characterize than the small molecule, or we can say chemical drugs. So with this, I want to give you some examples of biological drugs to make you understand that there's a therapeutic protein. There are several, and I can give you one example as filgrastim. So, and the other one is monoclonal antibodies. So the other one is the monoclonal antibody. The example is adalimumab. So dear attendees, please note that. So anything that ends with the name MAB, that is called monoclonal antibody. So we have a lot of monoclonal antibody nowadays, lot of biosimilars. We have a lot of therapeutic proteins with us now. So this is to give you some example so that the students can easily understand. So sometimes the students may have some eagerness to what extent this filgrastim or maybe the adalimumab may be used. That means these are the biological product. It's not biosimilar. These are the biological product, original biological product. So how that can be used and what extent that can be used in healthcare system. Just see. So this filgrastim injection that is used to treat neutropenia. So neutropenia is a condition where the patient will have low white blood cell because of its cancer. So you know, any person having cancer, to treat the cancer, they have to use some medicines. So that medicine generally produces a side effect, that is low white blood cell. That is a side effect. To treat the side effect is biological drug. There's filgrastim that may be used. So this is the usefulness of this biological drug, filgrastim. And we have another one is called adalimumab. It's a monoclonal antibody which is used to treat a variety of arthritis. Of course, students may be knowing that that is very difficult to treat arthritis, and we have several biological drugs nowadays to treat arthritis. One such what do you call this biological drug is the adalimumab. So this is original biological drug. So students, now we have to know what is the meaning of reference product. Why I'm telling all this, these are the interrelated ones. So once you know the meaning of biosimilars, you should also know the different terminologies with related to the biosimilar, and one such is the reference product. So reference product is nothing but a standard. So in case of TLC, we'll have a standard drug, and we do some extract TLC and we are comparing. So similar to that only, just for the for making an understanding, that's all. So reference product is a standard product, I can see in a rough word, and this is a biological product. So biological, so original biological product. So this product is already available in the market, approved by different agencies. It may be FDA or maybe EMEA, anything, any agency, they approved it and it's available in the market. And using this as a standard, that means this is a biological product. Using this as a standard, a copy is produced. That is called the biosimilar product. And this biosimilar product and the reference product, those to be compared. That means the biosimilar product should have similarity with the reference product. So that you have to keep in mind. So both are biological product, and one is copy, one is original. That you can keep in mind. So the reference product is approved based on, among other things, a full complement of safety and effectiveness data. That means reference product is already available in the market, so that is taken by the patient, so that there's a data, effectiveness data is available, safety data is available. So we use these data for comparison with comparing with the biosimilar product. That means the biosimilar product is expected to have same safety data and effectiveness data. And a proposed biosimilar product is compared to and evaluated against the reference product to ensure that the product is highly similar and has no clinically meaningful differences. So attendees, please note that. So here, the biosimilar product is very similar to the reference product, but sometimes it has certain, some differences. So that differences should not be clinically meaningful difference. Should not be. Then that means there is no clinically difference between the biosimilar product and the reference product. Means it's very similar, but there are small changes, and you must know what that change is, and that should be established or characterized. So this is the meaning of reference product. Next is interchangeable products. It's a very, very simple one. Interchangeable product is a biosimilar product only, and it is very close to the original product. So there is no problem at all. But if that biosimilar product is approved as an interchangeable product, then that may be substituted for the reference product by a pharmacist without the intervention of the prescribing healthcare provider. So sometimes what happens, these biological products are, it's a costly product in fact, and this will be substituted or be prescribed only by the doctors in the hospital. So you have to go to the hospital, meet the doctor, then he will make the changes, or we can say substitution or change of the drug, anything. So it is his wish, he will be doing. But in this case of interchangeable product, means biosimilar products, if it is approved as interchangeable product, then the patient need not go to the doctor, but he can go to the pharmacist, and pharmacist can able to substitute a biosimilar product with respect to the original product. So that there are two things here. He need not, means the patient need not go to the hospital, and doctor need not meet the healthcare provider, but there may be a substitution by the pharmacist himself. So this is known as interchangeable product. So there is another term which is known as bio-better. So bio-better is a controversial term, I can say. Bio-better is a controversial term with respect to the biosimilars. Why I'm telling you? Before that, just I want to tell you the meaning of bio-betters. Bio-better refers to a biological product that is same class as an existing biopharmaceutical, means it's a biological product only, but it's not identical. Just keep in mind, it is not biosimilar, means originally bio-better is a biological product that is expected to produce, means the company expected to produce a biosimilar product, but in fact, it has an improved property. So later on, the company have found that something good, better than the original product. So that means it is not the biosimilar product, but it's an improved version of the original biological product. Means it's another biological product, but it's a better product. So sometimes we call this as a controversial. Just I'm telling you. So biobetter will show improvement in one or more attributes, means qualities or properties over the original drug. For example, so the biosimilar product only, in fact, is initially produced as a biosimilar product, but later on it has been come to know that it is better than the original biological product, so that it is another biological product, and it has certain good quality. So what the good quality may be? So good property, it may be showing the less side effect, that's one, or it may be showing fast action, or it may require to have lower dosing. So anyone, if it is better, then it's called biobetter, right? So biobetter means it's another biological product, but it is produced based on an original biological drug. So just we want to make a copy, but this copy is having better activity or something, a better property, you can say. So it becomes now as another biological product. It's not biosimilar. It is not biosimilar. It's another biological product. How it is? It may be having less side effect. It may have some faster action, or it may require lower dosing only. So that may be the reason this is called biobetters. So biobetters are considered as a different active component, means different means another biological product when compared with the original product. With this, I can give only one example here. So there's a monoclonal antibody which is known as obinutuzumab. This may be difficult for you to pronounce, obinutuzumab. So this is monoclonal antibody. This is considered to be better than rituximab. So rituximab is another monoclonal antibody. So when compared with these two, the obinutuzumab is better. So we call biobetter. So initially I told you this is a controversial term. So why it is controversial term? That means we can use the word biobetter, there's no problem at all, but there are so many researchers, they don't want to use biobetter because we are comparing with another drug. So that another drug may be having very good activity in our body, so very useful drug, but simply just you're comparing and it will not be good. Means you're, the another one, it is good one, but still we are underestimating. Sometimes something goes like that only. So that is the reason there are many researchers, they don't use the word biobetters, and in terms of the usage by the people. So people always wanted a better drug. So that is in that angle. So this word may be used. So that's what I told is a controversial word. So many researchers, they don't want to use the biobetter. So why this is used? So generally this is used by the company to make money. That's all. This is only advantage for that company only. So here you see Genentech. So they produced obinutuzumab, and of course the same company also producing rituximab. So they only make that something is better than other one, and they call as biobetter. Okay, so dear attendees, I hope I made understanding of those terminologies, and I want to go for the advantages. There are several advantages of the biosimilars. Of course, the first advantage is the cost reduction by competition. So many of you know that the biological drug has, it is not cheaper, it's a very costly drug. And if you produce biosimilar drug, and these biosimilar drugs may be available cheaper. So that is the reason biosimilar drug should be available in the market. So the main reason is that is cost reduction. So biological medicine are costly and consume large portion of public and private drug spending. It means you go to the government hospital, private hospital, anywhere, the cost of the drug is more, so that you'll be spending a lot of money for that. In most cases, biosimilars are less costly than the innovative drug. So that means, of course, maybe more than 90% you can see the biosimilar drugs which are available in the market are cheaper than the original biological drug. Why? Why so? Why it is cheap? How it is available cheaply? There are few reasons. Because biosimilar drugs require few clinical trials. So we need to have only few clinical trials. The time, the money spending on the clinical trials are very, very vast. So you are saving a lot of money in that. And the second reason is that the cost of discovery and initial development is not needed. So you are copying another drug. Means for copy, you need not have the initial cost of drug discovery and development. So these two are not there. That means again you are saving lot of money for that. Because of that reason, you are able to produce so that's a biosimilar drug, and you can sell cheaper. So these two are the main reason. Not only that, there's additional competition might help explain the cost differences. So there are few companies, they're able to produce same biosimilar products, means based on one original biological drug. That means few biosimilars. If it is produced by different companies, then there will be a competition takes place. Then the company may reduce the cost of that particular drug. So because of a competition, there may be a cost reduction. So I can also give one example, which was from Europe. Biosimilar medicines offer potential economic benefit to the healthcare system. So this cost reduction, I told you. So there is one example from Europe. In Europe, the introduction of biosimilars has reduced the prices for biological drugs. For example, in October 2018, 80% price reduction on the monoclonal antibody Humira. So Humira is a trade name, brand name, which contain adalimumab. So that is a monoclonal antibody. So after the introduction of several biosimilars, means biosimilars of adalimumab that was introduced in the market, in the European market. Because of that reason, the original biological drug, that means Humira, adalimumab, they have reduced the cost of this drug up to 80%. That means, say for an example, if it was 100 rupees, now it is available for 20 rupees only. So that much of reduction is possible because of the introduction of the biosimilar drug. So everything is useful for the public. So with this, having multiple biosimilars in the market will break the reference drug monopoly and help bring down the cost. So that with this, we can bring down the cost, and we can also break the monopoly. So monopoly in the sense, adalimumab is the original drug, the brand name is Humira. It was the monopoly. So later on, a lot of biosimilars have been introduced in the European market. So because of that reason, the cost has been reduced. So attendees, please understand that the main reason of going for this biosimilar development for reducing the cost of the original biological drugs only. So there are several other advantages also. There's a competition and innovation. So I told you there are several companies, they produce a biosimilar product. So there'll be competition occurs, so that the price of the biosimilar products will be cheaper. So that's one thing. And not only that, there'll be competition occurs between the companies. And because of that reason, there may be innovative product development also possible. Then the third is enhanced patient accessibility. That means the patient will have accessibility to the biosimilar or biological product. So biosimilar medicines may improve patient access to the biological. So many patients who wanted to undergo treatment with the biosimilar products, of course, they will be having such kind of access. Then there will be treatment options also possible. Treatment options are very important for patients who have serious health conditions. So he can go for any treatment option if more number of biosimilar or biological products available in the market. Not only that, biosimilars allow some healthcare system to use innovative treatment for patients. Not only this, there may be innovative treatment with the use of biosimilars also possible. Then the incentive for innovation. So this is nothing but the funding to the companies. So pharmaceutical companies would be encouraged to invest more into cutting-edge R&D sectors to foster innovation. So of course, the pharmaceutical companies get funding from different agencies, then they are able to make some innovative product. And not only that, there are a lot of funding agencies, they can release the resources, of course, funding to the healthcare system, so that there may be a, funding new generations of the innovative treatment. That is also possible because of the development of the biosimilar products. Sunil, hello. I think you have to change some option, I think. So I'm unable to move to the next slide. Sunil, can you hear me? Can you hear me? Hello? 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 Hello?
Hello,
Can you hear
sir.
me? So
Hello.
sometimes I'm unable to move to the next slide. Very difficult to move to the another slide, next slide. So I think you have to change some screen setting, I think so. Can you hear me?
Hello.
Can you hear me?
Yeah, sir. Yes, sir.
Sunil? Sometimes I'm unable to move to the next slide because of some reasons, I don't know, but I have to change the setting, I think so. I'm not sure. Okay.
So can you move the slide, sir?
It was difficult. Now I am able to move, but sometimes it was very difficult to move. Even pressing
So
some tongue times, I was unable to move. No problem. I'm able to move now.
okay, sir.
Okay, right. So, dear attendees, there may be a question in you that why biosimilar products are developed now? Why this becomes a hot topic nowadays? Of course, there is a big reason. I tell you, patents on some biological drugs that have run out already or reaching to the end of their term, which is why different pharmaceutical companies are making biosimilars of these medicines. So attendees, please note that earlier I told you the patent of the original product means original biological product that should expire. After expiry only, it is possible to produce biosimilar product by the different companies. Okay, so keep in that mind. So when patent expire on biological drug, then various companies are allowed to make these medicines. That means you have to wait till the expiry of the patent of biological drug, not biosimilar product, biological drug. So an approved biological or biosimilar is expected to have the same safety and beneficial effects in patients as an existing biological medicine. So now is the time. What time it is? Now is the time where many of the brand of original biological drug patent expired already are going to expire. So this is the right time to produce biosimilar product by the various companies. So because of that reason, it is possible to produce, means make money also. There are two reasons. It is possible to produce biosimilar product, one thing, and because of that reason, you can also produce, means you can also make money. So these two are the main agenda of that company now. So now is the time because many of the product, patent got expired. That's one thing. And if you see how it has been approved, a biological product and biosimilar product that is expected to have same safety effect and beneficial effect in the patients. That means it is not a substandard drug. Biosimilar and the original biological drug both are having same activity effect in the data. Moreover, these products, maybe the original biological product or biosimilar product, these are approved by the same regulatory agencies and manufactured following the same high standard qualities, so that there is no difference at all. There is no question of substandard or low quality drug. So attendees, I can give you an example. I mean, a few examples of, the biologicals, the patent expiry in the US. Just to keep in mind, this is the US patent expiry. There are few drugs up to 2020. You can say there are 13 to 14 drugs. The expiry of, these biological drugs, these are the original biological drug. This patent got expired and few is going to expire maybe 21, 23 up to 28. So please note that there's a US patent expiry of this etanercept up to 2028, but there are biosimilar drugs are available for etanercept. So just I will give you maybe later on in the next few slides. I'm going to talk about this biosimilar product. Right. So what is the current status of the biosimilar products? So I'm going to talk about 3 different regions. So regions in sense, one I'm talking about the USA. The first one supposed to be on European Union because they were the first one. They means the European Union countries. They were the first to produce, means the approved biosimilar products in 2006. And later on, I'll be moving to the USA and what is the current status in India. So these 3 regions or 3 countries I'm going to explain to you. The first one is the European Unions. omnitrope that contains somotropin. So somotropin is the biological drug, means biosimilar drug in this case, right? Was the first product approved in European Union as a biosimilar in 2006. That means somotropin is biosimilar approved in the European Union by the agency, European Medicines Agency in 2006. So sometimes students may be knowing what is the European Union. European Union are the European Union countries, I can see. There are 27 countries right now under one union is known as European Union, European countries. And they have a common agency, which is known as European Medicines Agency. They are the one for them common, means maybe France, Germany, and several other countries. They are the one which comes under the European Union. So right now, at present, European Medicines Agency has recommended 64 biosimilar product with the different classes. So different class in sense, I made it here as 8 different classes. Human growth hormone, the example is somotropin. This was the first one which was produced, approved in the European Union. And second is granulocyte colony stimulating factor. So the best example is filgrastim. And attendees, please note there there's a suffix AAFI. So I talk about this maybe later on. And there is a erythropoiesis stimulating agent. It's called E14 alpha. Again, there's a suffix. Insulin. So the main one is insulin glargine. Then follicle stimulating hormone. There's folitropin alpha. So this Wavalip is the brand name. Okay. So a parathyroid hormone. It's called teriparatide. Brand name is Terosa. Tumor necrosis factor inhibitor. So infliximab. So there's a suffix, 4-letter suffix always here. Then there are several monoclonal antibodies, and these antibodies, which is given here, which is based on the rituximab. So rituximab is a monoclonal antibody, original drug. So based on this, there are a lot of biosimilars products are produced, which is names as CT, P10, GP 2013. There are several. Sometimes the name may be changed later on. There are several antibodies, the monoclonal antibodies are produced based on the rituximab. So what it says from this slide is that there are several biosimilar products that are already available in the market. Now, I'll give you some table in which there are a lot of biosimilar products with the product name, that is the brand name. Just I'm giving you here. So if we have a product name here, we can call as a brand name. So we have E14 alpha. It is used for anemia, cancer, and different categories, therapeutic area, which is given here. Then you have filgrastim as another biological drug. It's a biological. These are the biological drug, and here the product name is the biosimilar product with the brand name. That means Absimad is a biosimilar product sold in the form in the name of what in this name. That means the trade name, the brand name. This contains E14 alpha. So that means it's a biosimilar product. So we have filgrastim, adalimumab, so which is produced by different companies. So one is Amgen and Pfizer. So similarly have another 2 slides. I'm unable to move. So here I have a lot, and I already told you, dear attendees, etanercept. So I talked about this. So the etanercept biosimilar is also available here, only in European countries, not in USA. So Benapali, of course, here also, RLZ. So these are the different, the company brand name that contain etanercept. But the expiry, the patent expiry of etanercept in USA is 2028, but in European Union it is available. So there are a lot of biosimilar products are available in the market, and with the uses also, just I gave it here. So etanercept that is useful for different arthritis conditions. So we have rituximab biosimilar, filgrastim. So this is another one which is also available as a biosimilar product. And dear attendees, I want to give you an example. Means I can tell you the difference between filgrastim and filgrastim. So filgrastim is the biosimilar product means Giorgio, which contain filgrastim is a biological product, a biosimilar product. Of course, filgrastim is the original biological product. It is used for a variety of purposes like cancer, neutropenia, etc. So based on this, another product that is produced, it's known as pegfilgrastim. So PEG means PEG means polyethylene glycol, then filgrastim. So there's a polymerized filgrastim. That is the only one difference. Otherwise, that becomes the another biological drug. It's not biosimilar product, another biological drug. And for PEG filgrastim, there's another biosimilar product is the Udensia. Of course, there are different trade names. So this is the brand name that contains PEG filgrastim. So from this slide, we can come to know that there are a lot of biological products for which biosimilar products are existing and produced by various companies. So what is the current status in USA? Okay. But when you compare with the European Union, the USA, they have introduced the biological products in 2015. So after maybe 9 years later, the USA has introduced the biosimilar products in their country. The first one is the filgrastim with the suffix S and D Z. So it is in the name of Zerzio. That is a brand name. S and D is Z. That is indicating the suffix of this filgrastim. And by this way, it is possible to know that filgrastim is a biosimilar product. So earlier there was a confusion in the public, in the market, that which one is the original biological drug and which one is the biosimilar product. So there was a confusion. To remove that confusion, the USA introduced such kind of the nomenclature. We can see the nomenclature system. Sometimes they have some 4-letter suffix, 4-letter suffix. So that means S and D is Z in this case. So one can easily identify that this is a biosimilar product. In such a manner, there are a lot of the product for which the suffix is given, that indicating the biosimilar product. It's not biological product. It is a biological product in fact, but it's not the original biological product. These are the biosimilar products. In that manner, the nomenclature has been changed. Right. So this filgrastim S and DZ is a biosimilar product. It is based on filgrastim only, which is produced by a company in the name of Neupogen. So the FDA considers a product biosimilar if it has same mechanism of action, route of administration, dosage form, and strength as the original product. So please note that attendees. So the biosimilar product and original biological product that has similarities. What are the similarities? You can say it should show similar mechanism of action. There is no difference in the mechanism of action. Okay. Route of administration, of course, except the oral route, maybe any other route, it should be same. And dosage also should be same, and even strength also same. Everything should be same. Then we consider that's a biological product, biosimilar product. If any change in this, then it's not a biosimilar product, maybe biobetter, or it may be called the another biological product. That means what? A biosimilar product must have the same clinical indications for use as the original product. So this is also a condition in case of US FDA. So original biological product and biosimilar product which is produced in the USA, that should show same clinical indications. So this is a very important point to be noted in this case. In this case, the original biological drug filgrastim, which is produced and named as Neupogen, it's a brand name, which is manufactured by the Amgen company. So the another one, it's a biosimilar product, is called filgrastim SNDSZ. So suffix is manufactured by Sandoz, another company. Hence, it is a suffix, unusual suffix. It's a kind of nomenclature that is adopted in USA only. So biosimilarity does not guarantee bioequivalence. A very, very important point to be noted is that there is no guarantee of bioequivalence. So bioequivalence is possible in the small drug, chemically synthesized drug, but in case of biosimilar drugs, it's not bioequivalence, you cannot expect. And filgrastim SNDS means it's a biosimilar product that cannot be substituted for filgrastim, the original drug, without the direct approval of the healthcare provider. So that's what I told you. There's a product called interchangeable product. So interchangeable products, if approved as an interchangeable product, then it is possible to substitute it by the pharmacist himself. Otherwise, generally the biological product not to be substituted by the the pharmacist. So that is problem, because both are not bioequivalent to each other. So adverse effect profiles vary slightly using filgrastim S and DZ. So again, I just want to tell you that filgrastim S and DZ, so biosimilar product is not equivalent to the filgrastim. So that may be the reason it is having certain differences. So that differences, though it was characterized, but it is to be used or substituted only by the healthcare providers. Have same problem. Okay, dear attendees, here I can give you some examples of biosimilars that are available in the US market. Just you see, you can find only 19. As said now, maybe some 2 months back, just I prepared all these slides. So I could find only 19 biosimilar products that are available in the US market. So very, very less products are available because of several reasons. So one of the reasons is that the regulatory agencies are very, very strict in USA. So they simply don't approve the biosimilar product, but that may be one of the reasons why the biosimilar products in the US are very, very less. So what is the current status in India? So in our country, whether biosimilar products are available or not. So that we have to know. Yes, definitely it is available. Biosimilars are approved in the USA. It is available. And in India, we should call the biosimilar product as similar biologicals. We don't use the word biosimilar, but we use the word similar biologics. So all the Indian regulatory agencies, they use the word similar biologicals, not biosimilar. Okay, so it's available, but of course there are a lot are available. Near about 95 biosimilar products are available in the market. So these are classified in different categories like vaccines, monoclonal antibodies, of course insulin also. Then there are a lot of recombinant proteins. So classification that depends upon the company. The countries. So in this case, in India, so the regulatory agencies, they differentiate the biosimilar products in different categories like vaccines, monoclonal antibodies, insulin, and recombinant proteins. So now you may be interested to know whether the Indian biopharmaceutical company are producing the biosimilars or not. Yes, they produce. Many Indian biopharmaceutical companies engage in manufacturing and marketing of the biosimilars, not only producing, also they involve in the marketing of biosimilars. So Indian market for biosimilars will be expected by 2030. So in this year, maybe $35 billion. Means there's a huge potential. Means huge growth is possible in India. So there are a lot of companies, of course, we call multinational companies like Biocon, Glenmark Pharmaceuticals, Cipla, Lupin. Of course, there are so many companies, multinational companies are producing or focusing on the biosimilar market in India. So there are similar to other countries, we also have the regulatory agencies to address the issues and challenges associated with the development of similar biologicals. We have CDSCO, which means Central Drug Standard Control Organization. So CDSCO, in collaboration with the Department of Biotechnology, has developed guidelines on similar biologicals. So the name regulatory requirements for marketing authorization in India in 2012, and later on that was revised in 2016. Means all important guidelines are available with this, and these guidelines are addressing the regulation of manufacturing process as well as quality, safety, and efficacy. Means every information is available with this. It also addresses the pre-marketing and post-market regulatory requirements. So what is required pre-marketing, what is required for post-marketing, everything is available in that. And there's one condition which is given by this guideline is that it is essential for the company to conduct a phase 4 study with a minimum of 200 patients within 2 years of getting approval for marketing. This is one big condition which is required by the Indian agency. So you can ask me how many number of biosimilars available in India. So you'll be surprised to know this answer. So more than 95 biosimilar products are available in India. So please note that, dear attendees, more than 95 biosimilar products are available in India, either manufactured or marketed. So, but available in the market. So to name few, we have insulin, filgrastim, epotin alpha, adalimumab, cetuximab, and there are several monoclonal antibodies, even etanercept also available, and there are so many which are available. And of course, as I told you, more than, of course, 95 are available that has been reported. No. So having knowing the current status of different regions, including India, let us see how the biosimilars are developed. So maybe very complex process, but I want to go a little bit quick in this case. Biosimilarity of a reference product is established based on the totality of evidence. So this is an approach we call totality of evidence. So dear attendees, please note that biosimilar product is a copy of a original biological drug. That means you have to make a comparison. We have to compare the biological product, the original biological product, with the biosimilar product. Means this is called totality of evidence. Just compare, and you make that it's a very biosimilar to the original biological product. So you produce an evidence that's called totality of evidence, then that biosimilar product may be approved. So the bulk of the biosimilar development program is made of comprehensive analytical comparative testing as part of the justification for demonstration of equivalence or similarity. So please note that we have to produce a biosimilar product based on a reference product. That means we should compare biosimilar product with the reference product, and that comparison is called comprehensive analytical comparative test. So always comparing it, so that you justify that your biosimilar product is similar to the original biological drug. So you have to demonstrate in such a manner that the biosimilar product is very similar to the original biological product. So this is to be done with the analytical test only. So if both are same, say for an example, it's not exactly identical, it's not possible to produce identical biological product. So that is the reason. If it is very similar or similar, you can see, so that we can expect the same type of biological activity. You can expect, sometimes it will not also. You can expect very similar biological activities. So that is the reason the clinical part of the biosimilar product is very lean. So earlier I told you the biosimilar product require very little clinical part. Why it is? Because we will be already comparing these two products, and we establish that the biosimilar product is very similar to the original product. So if that is means the biosimilar product will have very similar biological activities with the original biological product. So that means you need not do much clinical part. So the clinical part is very lean in this case. And even then also there will be a small uncertainties, we call residual uncertainties will be there. So that should be addressed, that should be characterized. So characterized in the sense that uncertainties should not produce any kind of side effects or any kind of other activities, right? So here such a very complex picture, but I want to make you a little bit easy to you. So first you have to produce a cell line, right? So just you have to produce a line, cultivation and the production. You produce a cell line, and you have to optimize the cell line. Just you do the critical quality attributes. So CQA is here, critical quality attributes. Just you check it. So and from this you'll be selecting only one clone. So you select a clone. So just you preparing a lot of clones, and you will be selecting one clone. That's called one critical. What a critical quality attribute, just you selected. From this, whatever you have selected, that is to be cultivated. So that means you have to go for the preparation of the master cell blank. From that, you have to prepare your working cell blank. Then you go for the expansion. So this is a biotechnological way the product is produced. And you go for the production of the particular protein. So just keep in mind, all the biological product are the protein product. So you're producing protein here. Then after producing a protein, this is a mixture in this case, then you have to filter it, and you have to purify with the use of chromatography technique or any other technique. You can use any other technique, no problem. Depending on company, they use different technique for purification, and you compare the reference product and biosimilar product. So these both should be same. If both are same, we can say this is biosimilar. If it is not same, it is not biosimilar. So you should get a similar, the properties, you can say, whatever may be the property, that should be very similar. So if it is similar, if we are able to find that it's a biosimilar product, then go for the formulation, fill, and you prepare a finished product. So this is the way generally a biological product is produced. Of course, even biosimilar products also produced by this way. Attendees, I have a little problem. Maybe the setting is not good. I'm unable to move to the next slide. Now, dear attendees, the same thing only, just I want to give you a little more explanation. There are different stages of biosimilar drug development. So we have to go for the analytical, then go for the preclinical studies, then go for the clinical studies, then the phase 3 clinical studies. Of course, we have to consider phase 4 also maybe later on. So this is the way development of biosimilar process will go on. So generally 4 stages. So analytical stage, that's a bigger one. Preclinical stage, small. Of course, clinical stage, and these are very small only, just you can see. So there will be extensive physicochemical characterization to establish sameness of the biosimilar product with reference to the reference drug. So the molecular structure of the biosimilar product and the molecular structure of the reference product, that should be very similar. So you have to establish it. Then you have to move to the clinical part, that means preclinical part. Again, you have to use certain in vitro technique or in vivo technique with these certain animals, and you find the mode of action, and that should be very similar to the reference drug. Means biosimilar drug and reference drug should have similar activities. So similarly, you go for the pharmacokinetic studies and pharmacodynamic studies in human. In this also, you determine the bioequivalence. That means the biosimilar product and reference product should work in the body in the same way. There shouldn't have any difference in the way. There should be a, the similar way of activities produced by the biosimilar products and the original biological drug. That means reference product. And in the clinical study, phase 3, in that case, confirmation only. Just safety and efficacy is confirmed in the phase 3 clinical studies. Again, there's a comparison between biosimilar product and the original biological drug. Right. Now, this is a picture that says almost same only. First, you have to go for the structural analysis. Just you produce a data. In the data, what it says, biosimilar drug and original biological drug, means reference drug, that has similar structure, same. And function also, you go for the in vitro study, in vivo study, animal study, that also just make a comparison. The data should be very similar. Go for the clinical studies also, that should be very similar. But one thing much more important is that post-marketing. So this is to be means drug by drug case only. So this is also is needed nowadays, and that calls phase 4 study. Right. So as I told you, you have to compare the biosimilar product with the original biological product. So how we can compare? Just you can see, comparability of biological is the predictability of the relationship between structure and function. Of course, just you have to say that the structure is very similar to the original biological drug. So if the structure is same, you can expect the function is also same. So this is an idea, right? So in that case, what to do? Two biological that are highly alike analytically, means highly similar analytically. So structure if it is same, you can expect the same functions. That's what I told you. So sensitivity and specificity of modern analytical methods may discover appreciable analytical difference among the biologicals. So what happens, even though the structure is very similar, there will be small differences. So that differences may be identified with the use of certain certain analytical techniques, and that should be addressed, that should be characterized. So you have to characterize in such a manner that will not pose any threat to the human body or maybe the animals. So the macromolecules are characterized and compared at different temperatures, and the comparability assessment will be more reliable. So here one point is muted. So sometimes if you compare biosimilar products with the biological product at one temperature, that may behave similar. But if you change the temperature, there may be a difference in the behavior. So that is the reason you have to change the temperature. So then you characterize, then you'll get some data, and those data to be compared with the reference product, there should be similar data you have to produce. That means you should have the biological biosimilar product should have similar data. So what are the analytical studies to be conducted? That's one condition. And what you are going to establish. So you have to establish at least the structure of the biological product structure. So how you are going to establish the structure of the biological product? That's another question that may be arising in your mind. So an analysis will include the following: comparison of the biosimilar and reference product. So you have to have a biosimilar product, you have to have a reference product, just you are comparing. So you have to do a lot of analytical tests and you will come to a conclusion. So what are the analytical report you will get it? So you have to establish the primary structure of that particular biological product. That means amino acid sequence. So this is called primary structure. You will be establishing the amino acid sequence. Then you will go for the higher order structures. That may be the secondary structures, tertiary structure, or quaternary structures. So that should be established. Then there may be chances of modification. So we call a post-translational modification. So once you produce a biological molecule, there may be modification. I told you in the beginning only, the addition of certain molecule, or the molecule will have certain different shape. That means we call stereochemical nature, stereochemical modification. Sometimes we call, generally we call as a post-translational modification. That may be glycosylation, amidation, or deamination, anything that is possible. So that you have to characterize. And of course, product-related substances such as protein deamidation, oxidation, etc. These to be characterized. So these are the small changes that should be addressed. So during the analytical studies, biosimilar development must demonstrate that the analytical tests have the capability to detect differences between the biosimilar and reference product. Means, as I told you, you cannot produce exact, or we can say exact copy of the biological product. It's not possible, but you can produce very similar product. That's called biosimilar product. There will be differences. Okay, no problem. But in this case, we have to demonstrate in such a manner that the biosimilar product are very similar, but still there are differences. That difference is to be identified. Okay, for that we have to use a lot of instruments. So what are the instruments may be used? The instruments technique that are used, that should be based on the familiarity and efficiency. So first of all, you have to know how this instrument that can be operated by you, means familiarity, and whether it is efficiently detect the differences, whether it can be used efficiently to establish structure. So these you must know. So depending upon that, you must choose the different instruments. But normally we use the different instruments, namely mass spectrometry. So that means this will be attached with the LC, liquid chromatography. We call LCMS. That means HPLC along with the LC, means mass spectrometry, that can be attached. So we can also use capillary electrophoresis, whatever may be. And there are so many instruments that next slide just I'm going to teach. But whatever may be thing, we have to have a software program that is automatic, so that it is possible to process and evaluate the different properties of the molecule. It may be protein density, sequence variants, glycosylation profile, so anything that may be easily detected, but we have to have a software for that. That may be produced by every company, right? So to determine the higher order proteins, we have to have different, the instruments. Circular dichorism that may be used. FTIR, of course, many people you may be knowing it, and I have differential scanning calorimetry. This can be used, or this is a protein product, so that it can purify by size exclusion chromatography with a multi-angle static light scattering technique. So this is one kind of chromatograph technique. We can purify the components or protein molecule. We can use it. So just I gave the usefulness of the circular dichorism and DSC here. So this first one, circular dichorism, that is to investigate structural aspects of optically active chiral medium. It is mostly used for the biological molecules, structure, interaction with the metals, etc. So for this purpose, this is used. And then DSC, This is to characterize the conformational and thermal stability of biopolymers such as proteins, lipids, and nucleic acids. So conformational means size, shape, not size, shape. So as I told you, during the manufacturing of biosimilar biological product, there will be a different shape the compound may occur. So what is the shape that can be checked with the use of DSC? So there are a lot of other instruments also available to check it. So in this slide, just I gave you many more, and I think it's not required right now. You can find the primary structure, secondary structure, and of course see the potency. This is by the biological study. You can also find it. Then there are a lot of instruments that may be useful. Now, so just to show you some instruments or the pictures, diagram. You can say this LCMS instrument. It will be appear like this. There's a picture. Means the circular diagram, the working principle diagram. Of course, it's not that much required for you, but to make you understand, that's all. So you'll be doing a lot of analytical studies. So from the analytical studies, what you will find it? Just see, the results of characterization process may lead to one of 4 assessments regarding similarity. So when you do analytical test, you'll get 4 results. One, it is not similar. That means that product, whatever you develop, that is not biosimilar, that should be thrown, that's all. Or you should be going for reconsideration from the beginning. And if the product is similar or highly similar with fingerprinting like similarity, then that biological product is to be developed further. That means you have to go for the next step in the biosimilar development process. So that means you'll get 4 different sets: not similar, similar, highly similar, or highly similar with the fingerprint like similarity. So that means when you get similar, highly similar, that is the case, you have to go for the further steps, means other type of activities, and you have to go for the non-clinical in vitro function test. So in this, you'll be using either cell lines, in vitro cell lines, you can use it, or animal models, you can use it, anything. So these tests, whatever the tests you are doing, generally these tests should include all functionalities known to be active in vivo. That means you take a in vitro test. That in vitro test, whatever the results you are getting, that result is to be exploited in vivo. That means whatever the result you are expecting, that should be tested in vivo for confirmation. So difference between the biosimilar and reference product should fall within predefined domains. So this is another point to be noted. So you cannot expect the exact result, but you can expect some results very similar, and you have to have a limit we call predefined limit. Within that limit, your result should fall. Then you go for the pharmacokinetic study. So yeah, human pharmacokinetic serves several purposes. By comparing the pharmacokinetic data of a biosimilar product and reference product, the study data provide an important complement to the data from analytical studies in comparing likeliness of the composition of the 2 products, particularly with regard to glycosylation and other post-translational changes. So dear attendees, it may be very difficult to understand like this, but yeah, you'll be comparing it, that's all. The pharmacokinetic data of the original drug and biosimilar drug are compared, and if the data is very similar, you can say both are same. So already we have checked through the analytical test. In the analytical test, it's very similar. Even you do the pharmacokinetic studies in the human, the data should be same. If it's same, you can say that glycosylation also same. Other post-translational changes, it is not there. So that is a conclusion that you can make it. Not only that, this pharmacokinetic study is a sensitive method to support the likeness of the primary component of the clinical effectiveness. So when you do this one, you can say that all the data which are very same, then you can say that this is a primary data for the clinical effectiveness. So these are the data that can be checked. The third is very important. This will also tell you the immunogenicity of the biosimilar product. So I talk about this immunogenicity, maybe now. So the study allows systemic recording of the adverse events. So this also the side effect, this also that can be identified through the pharmacokinetic study. So regarding immunogenicity, of course, many attendees may be knowing that when you use biological drug to our body, the biological drug that produce antibodies. When it produces antibodies, and antibodies attack the biological product, and the biological product will not have any activity. That means no activity, just lost. So what to do? You have to make sure that the biological product will not have any immunogenicity. Immunity should not be there. So if immunogenicity is not there, we can say that the biological drug will work in our body. So make sure that the immunogenicity is not produced in our body. So this is the one which is to be considered very, very important in this case, immunogenicity. Now, pharmacodynamic studies. So this is very simple, of course, just comparison only. Again, I can say this is the another support of clinical studies. So many biosimilar drugs that do not have any suitable pharmacokinetic markers. Okay, so why this is to be done? So just see, pharmacodynamic parameters describe the action of the drug on the body. They describe the relationship between the drug concentration and pharmacological effect. That means if we give little quantity of the drug to our body, what happens? But increase the dose, what happens? So this is a study that should be done. And again, this is a further, confirmation of the activity of the biosimilar in our body. So human immunogenicity, as I told you, so this is to be recorded because differences in the immunogenicity that cannot be predicted entirely based on the analytical study. That means though you are making the lot of analytical studies in that you have understood that the both products are very similar, but still there is a small differences are there, maybe conformational changes, means shape of the molecule of the biological biosimilar, that may be little bit differences here. Because of that reason, that particular biosimilar product may produce some immunogenicity. So that is the reason human immunogenicity study should be done. It's very, very important, always to be done. So the study should be integrated into the pharmacokinetic studies. As I told you, pharmacokinetic studies also to be compared. Again, this is appropriate because anti-drug antibodies may cause changes in the pharmacokinetic data. So that is the reason immunogenicity study is to be carried out. So clinical studies, of course, I told you it is required, but in smallly. Regular clinical studies for the biosimilar products, but sometimes this is considered to be another unnecessary inheritance from the development of novel drugs, because this also takes time, permission, approval, so many. So that is the reason this is to be considered as an unnecessary work. Inheritance means it delays the development of the biosimilar product. But however, there are different, phase 0, phase 1, phase 2, phase 3. So what happened? What is the approach time that to be taken? Of course, that depending upon the company, depending on the product development, that difference will be there. But still, there are some time limits are given here. So overall, in this picture, I tell you the development process of the biosimilar. Just you produce complex protein, then you go for the non-clinical studies, then in vivo studies, in vitro studies. There are several data that you have to compare it, and finally you go for the regulatory approvals. So everything, every data, so whatever you produce with these tests, that should be given to the regulatory approval. So there is a regulatory agency. You have to submit all this data, and later on you'll get the approval. So dear students, we will find the challenges. So what are the challenges during the drug development or marketing of the biosimilar product? Right. So demand of biologicals is increasing in many healthcare sectors for small, when compared with the small molecules. So many people, they wanted to use the biological drugs. So the demand is increasing. Biosimilar landscape complications associated with the regulatory, manufacturing, biological test and marketing, that is concerned. Of course, we will have a lot of troubles also, right? From the producing or manufacturing, we do a lot of biological tests. Of course, we need to have big, very cost instruments, very difficult. I will need to have certain people who are good in operating those biological technique, and even marketing also. So there are a lot of problems we'll have, right? So these problems generally that can be, overcome. How it is? By outsourcing. So this could lead to the cost-effective development while minimizing some challenges. So there are a lot of challenges. Some challenges may be minimized, overcome by outsourcing. That's one thing. So all can be overcome with in-depth knowledge. So one should have the in-depth knowledge during the biosimilar drug development. So some company should know about the region. So whether in that region biosimilar products are available or not, if it's available, whether that can be sold to the public or not, whether the public will accept or not. There are so many reasons, I tell you. So they have to identify the region and early strategy planning. At the earliest, they have to produce and sell it, and they should have the effective communication with the regulatory agencies. It's a very, very important one. Sometimes the regulatory changes may be there for each and every time. So depending on time, the product, there will be changes in the regulatory agencies. So they have to have a communication with the regulatory agencies. As per them, the company has to produce the biosimilar product. So these are also the challenges only. So the successful development and commercialization of the biosimilar products that require business strategies that integrate appropriate clinical design and regulatory compliance. That means one person or company should know the regulatory process, how to get the approval for the marketing, clinical design, of course, each and everything. And they have to have the updated knowledge on the regulatory problems. So in the beginning only I told you it's very difficult to produce two different biologicals by two different manufacturers same. It is not identical to each other. So there will be a variation. So what may be the variation? I already told you. The variation may be post-translational modification. It may be glycosylation, maybe phosphorylation. Means addition of phosphor, addition of sugar. So not to be confused much. Glycosylation means addition of sugar. Phosphorylation means addition of phosphor. Carboxylation, addition of carboxylic group. So there may be a difference. So because of these differences, there will be a degradation, there will be aggregation, there may be deamination, there may be a lot of changes may be taking place in the biological molecule. Because of these changes, there will be a change in the shape of the molecule. So conformation changes means shape of the molecule will be different. Because of the shape of the molecule is different, because of these differences, post-translational modification, there may be chances of immunogenicity. So these are the different challenges that should be optimized. Right. The next challenge is access to the sample of the reference product. So please note that we have to have a reference product, standard product. So when the standard product is not available, what to do? It's very difficult to compare. So this is a very, very big problem. So access to the sample product is very difficult sometimes, and which we need it for the drug testing and development. So before a company may market a biosimilar, it must demonstrate that this product is highly similar to the reference product. We need to have a reference product for the comparison purpose only. So such comparative testing generally necessitates access to samples of a reference product. Of course, we need to have a reference product. So companies have implemented self-imposed restricted distribution system, means reference product manufacturer, they have implemented self-imposed restriction. So they don't want to sell to the other companies. They don't, means the other company who wanted to produce biosimilar product, they want to have the reference product which is not available to them, so that there will be a great delay in the development of the biosimilar product. So by withholding access to sample, the brand name company is generally able to prevent or delay a biosimilar product developer from conducting the required testing. So this is the reason the biosimilar product development will be delayed, so that the original biological product innovator company, they still play a monopoly and they will make money. So this is another challenge. Then challenge to the patient and prescriber. So we need to have a good education. So what is the education? Patient education. The patient should accept it. And physician, means doctors generally, the prescriber, they also should know about the biological product, whether available in the market, what kind, how dose, everything. And the pharmacist, we have a role and we should know. Of course, I told in the beginning, if you know the regulation of the interchangeable product, yourself you can substitute. So you have to have a knowledge. So the knowledge on biosimilars is important. So all the doctors, pharmacists, patient should educate or enlighten the knowledge and convince the benefits of switching to the biosimilars. So when using the smaller molecules, of course, it is having some activity, but sometimes the biological drugs will be having better activity, or sometimes this is the only choice, and it should be convincing, means pharmacists should be convincing the physician also, and the patients also with the use of biological drug. Then regarding interchangeable drug, I already told you the pharmacist, only one person, he can change or substitute reference drug to the patient. This is also possible. So another challenge is that the rare disease. So the disease is very rare. So the patients are not available. So the drug development may be very difficult. So this is also another problem. So for this, just I want to give you some example also. Biosimilar developed for orphan drug in the sense rare disease, the drug for the rare disease. This face some clinical problems, the practical problems. The difficult to obtain a large and enough heterogeneous population for phase 1 to phase 3 clinical trials. So difficult to find the people, and manufacturing enough batches of biosimilars to run batch to batch variability studies. This is also difficult, and because of that reason, it becomes a big challenge. So I want to give you a rare disease example. Sometimes the attendees may ask, what do you mean by rare disease? So I can give you 2 examples here: acute lymphoblastic leukemia, digestive system melanoma. So these are the, of course, there are hundreds of rare diseases, but I want to highlight only 2 of this here. So attendees, what is safety of the biosimilars? I already told you the biosimilars are safe. There is no problem at all. But I want to give you some more information with regard to the safety of biosimilars. Because biologicals are administered parentally, the devices are recorded generally. Why? This is not to be taken orally, just to keep in mind. We need to have certain device. Through the device only, the biological is to be administered to your body. Right? So the design of the device is the patent of that particular company. So that means it is also patentable. So the biosimilars will have a different device, not only in appearance, but also potentially the function. So there's a device, the device will have a different design, appearance, and function. So every company should produce their own device that will be different in their appearance, function, and design also. So this is patentable. So what to do in this case? We have to have enough knowledge in this to administer a particular biosimilar into the body of a patient. That's very, very important in this case. So because of the uncertainties associated with the use of biosimilars, pharmacovigilance is also important. This is also a very, very safety point of this biosimilars. We need to have a pharmacovigilance, of course. Moving to the diversity in their structural complexity and indications, so safety will need to be considered on a drug by drug basis. So for every drug, the pharmacovigilance is to be done. There should be drug by drug basis only. So this is regarding the safety of biosimilar drug. So attendees, you'll be asking what will be the feature of this biosimilar, whether the feature of the biosimilar drugs will be available or not, if it's available and what extent it's available. Yes. So it will have a great impact even in industries also, even for the patient, hospitals, anywhere. It will be available in the future also. Let us see what is the impact on the industry. So biosimilar products will have a significant impact on the pharmaceutical industries, not only in the US and Europe, but throughout the world. So throughout the world, it will have some impact. So due to the increased competition with the accelerated approvals of biosimilars and the rapid introduction to the marketplace, market conditions will have to evolve and adapt. So in the future, there are a lot of biosimilar product will be available. So sometimes it will be available in the market very fast, rapid introduction, that is also possible. So what to do? The market should evolve itself to adapt. It should accept it. So this is a market that will remain in focus in this billion-dollar industry. So this market we call as a billion-dollar industry. So there'll be a change of the industry forever. So the face of the industry, the pharma industry may be changing forever. So when compared with today's, there'll be a change will be there. Then so biosimilars with complex structure, of course, that is already available, and also in the future there'll be a multi-subunit, extensively post-translationally modified, and lipid-containing products will also be available. So these are very complicated, and that may raise new complications and concerns, but it is going to be available in the market. So we have to know, and we have to have enough knowledge on this as a pharmacist. A number of new companies have recently entered in the biosimilar development scene, and They are marketing fast progress. This also I told you. So due to the introduction of biosimilars, the key stakeholders like prescribers, pharmacists, nurses, patients, everybody will face many challenges. There are a lot of challenges. We have to accept it. We should know what are challenges, and accordingly we have to try to solve those challenges. The acceptance of biosimilars by the general public and their ample use in the healthcare system is another challenge. So it is available, and if it's not accepted by the public, then there's a problem. If it's not accepted by the healthcare provider, it's not a problem. So we should educate the patients. We should educate the healthcare provider for acceptance of this biosimilar products. So these are different challenges. So how the drug that may be administered? The patients receive biological by injection only, by under the skin, we call a subcutaneous injection, or by intravenous infusion, and cannot take them orally. So please note that, attendees, we cannot take orally. If it's taken orally, what will happen? The biological becomes ineffective. The process of digestion breaks down the biological drug that makes it ineffective. So here in this picture, I can show you the different, the devices, the drugs produced by different companies. So I'll come to the conclusion part. So many biosimilar products are available in the market. Of course, it's available, no problem at all. But what is your role? Your role in the sense, it's our role. In fact, I am also a pharmacist, so our role. So we will have a big role in the healthcare system. So how? So it is your role that you have to handle the biosimilars in a suitable way. That means you should know the storage conditions, where to store, how long that can store. So any point related to biosimilars, you should know. So handling everything that comes from handling, so you should know. So you will have a big role in handling the biosimilars, including storage, the length of storage, etc. And your role is to educate the patients and healthcare team with the use of device, and also you can tell them the dose or any other information related to the biosimilar product. So it is your role that you should educate the patient, you should tell the patient to take the biosimilars in a right way. And also you should educate the healthcare provider. That means he may be the doctor, it may be the nurse, or any other person who related to the healthcare team, you should educate them. So to educate them, you should have the good knowledge. You should educate first, and then you teach others. So this is another big role. And you also have a role in pharmacovigilance, means any adverse reaction, if it's reported to you, and you should report to the higher authorities and to the company. And you'll have a lot of other roles also with biosimilar product. So dear attendees, I hope you have a little idea about now on biosimilar product, their uses, how their advantages, and how it has been developed or is going to be developed. So development process in general, what are challenge, etc. So with this, just I conclude my webinar. If you have any questions, please. Any questions? Sunil?
Sir.
Any questions from the student side, especially?
Sir, one of my student asking for one question, sir.
Okay.
What are the biosimilar products used for COVID-19 treatment?
I think that has been recommended by some company, that remdesivir, something like that is given. But otherwise, there is no specific drug. These are produced for different other purposes, not for COVID. But still, there are some people, they recommended some kind of biological product for COVID. Any other questions?
No, sir. No, sir.
Questions from the staff side? There is
Srinath,
no
sir?
chart. You could have given the
Yes, sir.
the charting option to the students so that they can put the questions there. Chart, group chart, no. No question, I couldn't find any question in the chart. Okay, any other questions? There are 89 participants and nobody's asking questions.
Sasina, sir.
Yes,
Yes,
yes.
sir. Yes, sir.
No. Okay.
Yes, sir.
Any-
Thank you, sir. My name is Srinath, sir.
Srinath,
Associate
please.
Professor. Yes, sir. Department of Pharmacy Practice, Sun Institute
Okay.
of Pharmaceutical Education Research, sir. Sir,
Okay.
now I am very happy for vote of thank for today's international webinar. So first, I would like to express a great thank you for our speaker, respected Sir Dr. N. Madhavan, sir, for this webinar, for sharing your valuable view, knowledge regarding about biosimilars, its current status and prospective updates, sir. Thank you so much, sir. In this topic, we learned very much knowledge, sir, like safety and effectiveness and reference products and interchangeable products, what is the bio better and what is the safety considerations. So in that cost, what is the advantages like cost reduction by the companies, how basically it having the less few clinical trials, so it may helpful for the cost reductiveness and enhanced patient accessibility like that. And we learn many words like patent expire important for the biosimilarity of the drug, and FDA importance for the further steps like bioequivalence, adverse effect profile, and various effect may influence our FDA. And we learn much about the current situation of India, almost 93 drugs in bioassessor. Thank you so much for that. So we know about that CDCO like guidelines and manufacturing in the maintain the manufacturing quality, safety, and effectiveness. And so you are giving the very valuable information about the maintain of the total of evidence and comprehensive analyticals, what is the similarities and what is the uncertainties. So and CQA. So what we want to know in CQA, like the cultivation, production, isolation, and purification steps, and formulation, and fill, and finally finishing. There is a having different stages for checkpoints, sir. We learn about more about that stages like analytical and preclinical studies, clinical trials in the pharmacokinetics and pharmacodynamics, and clinical phases, how it may helpful, and biosimilar developments and analytical studies, how may helpful like that, sir. And we learn and we know most about that instrumental techniques, how may helpful, familiarity and efficacy most important, and how to maintain the software programs also may helpful for the biosimilarities, sir. And determinations of higher order protein structure. So here protein structure is the most important. You said, sir, safe with the DSC, and we know about that. We learn about that comprehensive analytical studies like the similarities. Oh, sir, and the today topic we learn about the non-clinical in vitro function test, how we want to conduct, and how we will get the about the results like that, and enhance the pharmacokinetic studies, the clinical effectiveness and immunogenicity, sir. In that you are giving the very valuable suggestions like that, how to identify the adverse drug events, how to control, and you are giving the very valuable about the biosimilarities and anti-drug antibodies like that, and what is the role of the clinical pharmacist and pharmacist. This is the most important. Why? Because biosimilarity is the new drug, so the pharmacist want to educate the patient how to use and route of administration, what is the dosage forms. Sir, you told like that this is a specific devices for this drug. So if suppose we are giving the another route, it may not giving the proper pharmacokinetic effect, but sometimes it may causing to the adverse drug reactions in that. So want to maintain updating knowledge, sir. You tell like that is updating knowledge. So confirmly we will do updating, sir, and we will educating, and we are in this mainly want to report AR, and adverse drug reporting is most important for the studies, sir. You tell. And my way, sir, sorry for the inconvenience. So there is some systemic error, sir. Thank you, Winan. And on behalf of the organization committee, we're thankful for your fabulous participation, support that you have extended our students and to delegates for the making the grand success of the webinar. Sir, thank you one more time. In spite of your boys busy schedule, you have reported to the webinar within a short notice. Maybe you was effective, transparent with the commitment and dedication, sir. So your presentation on bisimilarities and its current situation and prospective approaches as disseminated knowledge embedded with modern trends and techniques. So we look forward to organize many more seminars, webinars under your eminent guidelines and the leadership, sir. Once again, thank you from Sun Institute of Pharmaceutical Education and Research, Kakupalli, Nellore. Thank you so much, sir.
Sir, 1 minute, sir. One person is hand raising, sir.
Yeah, somebody's hand raising. Okay, please. Question?
Oh, no, sir. 1 minute, sir.
అన్మ్యూట్ చేయాలి. అన్మ్యూట్ చేస్తే తెలుస్తుంది ఎవరని.
సార్ హ్యాండ్స్ రైస్ చేశారు, అందుకే అన్మ్యూట్ చేశాను.
సరే, మంచిగా చెప్పినారు సార్. సార్ హస్ గివెన్ వెరీ గుడ్ డీటైల్డ్ this thing right from the beginning of biosimilar to bringing into the market.
Hello.
Okay, complete it, sir.
Yes. Thank you so much for Srinath and Dr. Shekhar.
Thank you, sir.
Thank you so much.
Mahadevan sir, thank you very much.
Thank you so much for inviting me. So
one Dr. Mohdin is there, pharmacognosy in KSC. I don't know which university, King, may not be King College, some other university is there.
Maybe.
Okay, sir. It is very nice to hear biosimilars.
Definitely, sir.
Thank you very much. Let us meet once again. So, by God's grace.
Thank you, sir. Thank you so much.
Give one more session for us. One or two more sessions in this academic year.
If you want in any other, department, I'm able to arrange. I'm able to arrange.
This year, we are very badly affected because of the COVID.
Yes.
Because
definitely.
of the COVID, we couldn't conduct 30% of the classes. We couldn't conduct.
Yeah.
So the only way is like this only. Not only
Yes,
that, when we ourselves are taking classes, our students do not join this much. When we say that when we say that it is a national seminar, some outside eminent speaker is there, they participate.
Yeah, definitely.
So thank you very much. Let us hope for many more sessions from your side, sir. Thank you.
Definitely, sir. Thank you so much, sir.
Thank you, sir. Thank you very much.
Thank you, Sunil. Just I leave the session, okay?
Okay, sir. Thank you.
Thank you so much. Thank you.
Bye all. Bye all. Apa Rabi?